Hello everyone, I am Srijith and I am a PhD student in Department of Biotechnology IIT Madras, and I am here to talk on the research on exploring the metabolic plasticity of QTNs and their combinations using systems biology approaches. One of the major challenges in modern biology is to understand how the naturally occurring variation in DNA sequences can cause phenotypic variation in complex traits. The observed phenotypic variation can be either classified as Mendelian traits or complex traits. These Mendelian traits are monogenic in nature. For example, Mendelian disorders like sickle cell anemia and several other Mendelian traits include flower color, pod color, etc. Whereas the complex traits are polygenic in nature, where several genes interact at several levels and time to modulate the phenotypic variation. These complex traits are also called as quantitative traits as they are quantifiable. Most of the human diseases like cancer, diabetes, etc. fall under these complex traits. So when we see the distribution of these phenotypes, we generally see a normal distribution where the extreme end has the highest phenotype due to the presence of highest number of quantitative trait nucleotides. These are SNPs which are responsible for these phenotypes. Understanding how these SNPs contribute to phenotype can have several implications in the field of precision medicine, breeding programs and strain improvement, and also in understanding the adaptive evolution. Genome-scale metabolic models are currently used as an in silico tool to test various hypotheses of genotype-phenotype relationships. Recently, the role of variation or a SNP in enzyme coding regions have been studied by changing the Vmax of a particular reaction where the gene is causing the effect. But no studies have explored the role of genetic variation in transcription factors which are neither enzyme coding regions that can cause metabolic flux perturbations. Further, no studies have been done to determine how multiple variants interact at molecular level to cause these metabolic changes. Here in our study, we hypothesize that these interactions between the transcription factors can cause an activation or deactivation of a flux of a metabolic enzyme, thereby changing the metabolic landscape of the entire cell. In order to understand how these SNPs in the transcription factors can cause a phenotypic variation via the metabolic flux distribution changes, we chose yeast correlation efficiency as a model where RME1, RSF1 and IME1 are three genes responsible for high sporulation efficiency. Studies have also identified the SNP level changes in these three genes which are causing this sporulation efficiency variation. We try to ask these following questions. How do these variants individually and in combinations can affect the expression patterns of the metabolic regulators? Further, we asked how these variants individually and combination can modulate the flux compared to the wild type, thereby having a metabolic flux variation causing this correlation efficiency variation. We took the transcriptomic data of 16 QTN replacement panel from an already published study and did a differential gene expression analysis to identify the genes that are differentially expressed by these QTNs. Further, we did a PCIT and regulatory impact factor analysis to identify the co-expression changes due to these variants in each of the allele replacement strains. In order to understand how these QTNs have an effect on the metabolic flux distribution, we used genome-scale metabolic modeling in specific by integrating the transcriptomic data with the genome-scale metabolic model of yeast. This will help us to understand how these QTNs individually and in combination can activate or change the flux of a specific reaction. We used local Guinea thresholding and IMAT to reconstruct these 16 QTN specific metabolic models. Further, we analyzed for unique reactions in each model compared to the wild type, and further we did OPTGP sampler to characterize the flux of each of the reactions between the wild type and the QTN. Then we calculated the flux change for each of these common reactions and identified the upregulated reactions specific for that particular QTN and their combinations. These upregulated reactions that were then checked for subsystem enrichment using flux enrichment analysis in Cobra toolbox. Our differential gene expression analysis, by comparing the wild type with each of these allele replacement panels, we showed that each of the variants have a specific set of metabolic genes and sporulation genes that are getting differentially expressed. Further, we showed that the clustering of gene expression patterns show variant-specific modules. That is, for example, the REC8, a gene that is involved in recombination, is differentially expressed only when the RSF1 coding variant is in combination with the IME1 coding variant. This reveals the genetic architecture of metabolic flux remodeling in each of these allele replacement strains. By using the partial correlation using information theory approach and regulatory impact factor metrics, we identified the co-expression profiles of each of the allele replacement strains. Further, we calculated the connectivity of each of the metabolic regulators that are identified from the RIF analysis. The RIF analysis identified 7 metabolic regulators that are involved in amino acid metabolism, histidine metabolism, and biosynthesis of steroids. When we did a clustering of high, slow, and medium sporulation efficiency for these connectivity, we found that the UPC2, a gene which is involved in steroid biosynthesis, has the highest change in connectivity across variant combinations. Further, these results highlight that the changes in the gene co-expression network connectivity can modulate the metabolic flux in core meiotic and sporulation-specific metabolic genes, thus regulating the sporulation efficiency variation. From these analyses, we highlight the fact that several metabolic alterations happen in each of these allele replacement panels, needing the use of genome-scale metabolic modeling to further elucidate the molecular mechanism by which each QTNs can affect the metabolic flux distribution. As discussed in our approach, we use the local Gini thresholding and IMAT to reconstruct the variant-specific metabolic models from the genome-scale metabolic model. Further, we use the constraints related to acetate and glucose to recapitulate the meiotic conditions in yeast. By this way, we were able to reconstruct 16 variant-specific metabolic models which were named as M1 to M60, and each of these models have different reactions highlighting their metabolic heterogeneity. Further, we classified them as high, medium, and low sporulating models based on the phenotype. By comparing the common reactions between all these models, we identify the core metabolic network which are enriched for steroid biosynthesis, glyoxylate and dicarboxylate cycle, and pentose phosphate pathway. We hypothesized that each of these variants and their combinations can influence the reactions involved in this core metabolic network to modulate the sporulation efficiency variation. Further, we asked how the coding and non-coding variants present in the same gene can show phenotypic plasticity and metabolic plasticity. For this, we compared the models of IME1 coding, IME1 non-coding, and their combinations. By using the flux enrichment analysis for the unique reactions present in these models, we show that the presence of IME1 coding variant can have several reactions that are active, whereas in the presence of non-coding variant, many of the reactions are not active. This highlights the fact that the presence of coding variant can form a functional IME1 protein, whereas the presence of IME non-coding variant forms a non-functional IME1 protein. This was further highlighted in our genome scale differential flux analysis, where we compared the upregulated reactions. As seen in here, we show that IME1 coding and the combination with the non-coding have few set of reactions that are common between them, whereas IME1 non-coding, where the non-functional protein is formed, have a unique set of differentially upregulated reactions present in them. Using the differential flux analysis, we were able to show that Pentose phosphate pathway flux variation is a consequence of variant interaction. That is, by closely examining the models where the pentose phosphate pathway is getting upregulated, we were able to cluster them into two specific groups. We were able to see that the models where IME1 non-coding variant is present, we see a pathway flux which is comparable with the wild type. Whereas in the other case, the pathway flux is being upregulated whenever IME1 non-coding variant is not present. This shows the differential regulation of pentose phosphate pathway in an IME1 NC dependent manner. These are the flux distribution plots of specific reactions in pentose phosphate pathway. In specific, the glucose 6-phosphate dehydrogenase is a reaction that is first in a first rate limiting step of the pentose phosphate pathway. Further, ribulose 5-phosphate 3 epimerase reaction is a reaction that is involved in several downstream pathways which are essential for the production of nucleotides and amino acids. Here we try to ask what are the subsystems that are essential for efficient sporulation. In order to do this, we use the 5 models which are highly sporulating in nature. That is the one with all the 4 QTNs and 3 QTN combinations. By comparing these models, we were able to see that the one with all the 4 QTNs have 6 subsystems enriched which are upregulated, whereas the other models show a variable subset of this M1 model. This highlights the 6 subsystems that are essential for the efficient sporulation. Further, we were able to see that the glycerolipid and glycerophospholipid metabolism is present in all these models, highlighting the highlighting the importance of this pathway in sporulation efficiency. Next, we try to ask why this pentose phosphate pathway is specifically enriched in these two models, whereas not in the other models. We know that the pentose phosphate pathway is essential during the initial phase of sporulation because pentose phosphate pathway can produce nucleotides and amino acids required for cell division. So we asked what are the other compensatory pathways by which the other 3 highly sporulating strains can use to produce the nucleotides and amino acids. By closely examining the active reactions present in these models, we were able to see that autophagy is differentially active between these models. This led us to a hypothesis that during nitrogen starvation and non-fermentable carbon source, pentose phosphate pathway is generally active to produce nucleotides and amino acids required for sporulation. Whenever the pathway is not active, the yeast uses another compensatory mechanism by which autophagy, where it degrades the cellular components and recycle them for the production of nucleotides for efficient sporulation. By leveraging the genome-scale metabolic modeling, we were able to reconstruct variant-specific metabolic models for each of the allele replacement strains and showed the variant interaction can have an effect on the metabolic flux distributions. This highlights the importance of specific pathways in sporulation efficiency variation. Further, we were able to predict autophagy as a pentose phosphate dependent compensatory mechanism for yeast sporulation. We argue that further experimental validations are required to prove our hypothesis. However, our framework can be applied to understand the molecular basis of complex human metabolic diseases where multiple variants interact and contribute to disease phenotype. This will aid in the better prediction of the disease targets for therapeutic interventions. I would like to acknowledge my research supervisor Dr. Himanshu Sinha for his continuous support and encouragement throughout the project. I would like to thank my collaborator Dr. Neerav Bhatt and Pavan Kumar with the help in the modeling part, Systems Genetic Lab and IBSC members for their timely help, IIT Madras and MHRD for the fellowship, and finally the ISMB for the travel grant. I would like to thank the ISMB for this wonderful opportunity for presenting my work. Thank you.